All right. So day 4 of organic chemistry. This is our summative review, and all I really do in the summative reviews is I just kind of go back through the review questions that we had throughout our daily lesson, just to have them all in one place for you. Just as a quick recap, you'll probably still want to go back to those to actually be able to see the information as I talk about it. And I would definitely use those other videos to help with your studying. Okay. And so, of course, this is the order, the same order that we went into it. So, day 1 review. So, I asked you about the 4 organic molecules in living organisms. Just kind of take a second, think about that. I don't want to go over the answer to it because I want you to actually take time to think about it, write down your answer, come back and look at it. The 4 main organic molecules, we're talking about carbohydrates, lipids, nucleic acids, and proteins. Those are our 4 that we're talking about. Make sure you got it correct. What elements make up each organic molecule? What elements make up each organic molecule? Just give you a second. See if you can write this down. That way you don't have to keep pausing it each time. I'll just give you time to write it down. And you can just put down the letters. You don't have to write up the whole words as long as you know what they mean. So to make this process easy for you if you're kind of stuck, we need to make sure that we remember that everything has to have carbon, and out of those 4, all of them have carbon, hydrogen, and oxygen. So that's a good starting place with all 4 of them. They all contain carbon, hydrogen, and oxygen, except for nucleic acids. They also have nitrogen and a phosphate group. Phosphate. And proteins also have a phosphate group. I'm sorry, not a phosphate. They have a nitrogen group. A nitrogen group. But the carbohydrates and lipids only carbon, hydrogen, and oxygen. Now, the main element that must be there, carbon has to be there. Remember, deciding between that organic versus inorganic. What's the function of a nucleic acid? To store and transmit genetic information. The subunit is called a nucleotide. Make sure you know the 3 parts: phosphate group, pentose sugar, and the nitrogen bases. Make sure you remember those nitrogen bases. The differences between DNA and RNA: DNA stores genetic information, RNA transmits genetic information. DNA is a double stranded molecule, RNA is a single stranded molecule. DNA um, contains the nitrogen bases adenine, thymine, the T, cytosine, and um, guanine, while RNA contains the nitrogen bases adenine, U for uracil, cytosine, and guanine. And then DNA has the sugar deoxyribose, while RNA has the sugar ribose. Day 2. This is when we're talking about our energy molecules, lipids, and carbohydrates. Which 2 organic molecules are responsible for storing energy? Lipids and carbohydrates. What's another name for lipids? Anytime we think about lipid, we need to think about the term fats. Fats. Some examples of that would be like oils and waxes, and of course, fats itself. List 3 foods where you can find carbohydrates: potatoes, pasta, fruit, vegetables, rice. Those are delicious things. What's the main function of a carbohydrate? The main function is short-term energy storage, while lipid is its big deal is long-term energy storage. Lipids also function to create cell membranes. They're the key component in a cell membrane. That's called a phospholipid. They're also very useful in waterproof coatings because oil doesn't mix with water. Fats and lipids don't mix with water. and also insulation for basically to insulate warmth and, of course, cushion and protection. What are the four polysaccharides? Four polysaccharides. And remember, a polysaccharide is a carbohydrate. Our four polysaccharides would be starches. That's what's found in plants. That's how plants store carbohydrates. That's how they store the glucose. You also have cellulose. That's what makes up a plant cell wall. You have glycogen. That's how animals store glucose, and they store that in their muscles and liver because that's where animals need energy. We need it in our muscles. And then last one is chitin. It's found in a fungi cell walls like mushroom cell walls, structural component for mushrooms and fungus. What is the difference between a monosaccharide and a polysaccharide? A monosaccharide is the subunit of a carbohydrate, and that means one single sugar. And whenever we stick a bunch of them together, that's what makes a polysaccharide. A polysaccharide is many monosaccharides. Please don't forget the subunit of a lipids. I don't have that one on here. It doesn't look like is glycerol and fatty acid chains. Now we come down here to the last question is the difference between a saturated fatty acid chain and an unsaturated. Saturated would have nothing but single bonds on those C's, on those carbon atoms. They're unhealthy for you and they're typically solid at room temperature, while unsaturated fatty acids are liquid at room temperature. They have that double bond, so they get that kink in it, and they are healthy for you. Be like vegetable oil that you see in the cabinets, liquid, or like olive oil. And then, of course, last one was the one from last time's notes would be day 3, talking about proteins and enzymes. So some functions of proteins. Big one, regulate cell processes, maintain homeostasis. They are building blocks, or you could also say structural components. The subunit of a protein is an amino acid. How many kinds of amino acids are there? There are 20 kinds of amino acids, 20 different kinds in the human body. Which organic molecule is an enzyme? An enzyme is a special type of protein, and they're going to help out with those regulating cell processes and maintaining homeostasis. It does that because it's a catalyst. And a catalyst is what an enzyme is acting as because it's speeding up the chemical reaction by lowering the activation energy cost. And I had that graph on day 3's notes that you can look at where I kind of explained that out toward the end of the notes. What can cause an enzyme to denature? 2 key environmental factors would be changes in temperature and changes in pH level. Why are enzymes described as a lock and key? Because only a specific substrate, which would be the key, could fit into a specific enzyme, and that enzyme is the lock. And and on day 3's, I had a graph. I showed you some graphs of what that optimum rate would look like and how to read those graphs. Don't forget, you'll also need to look at that image of the enzyme and the substrate complex and make sure you can identify the substrate, the enzyme, the active site, and the products there at the end. So make sure you go back over your notes, look back over our actual day videos and not just this one, and come to class prepared for your test.